During vaccination, each animal is initially injected with a small volume of venom. 2 weeks later, it is injected with a larger volume of venom. Use your knowledge of the humoral immune response to explain this vaccination. So the beta cells are going to be specific to the venom reproduced by mitosis. So when the B cell that has a complementary receptor on the surface to the venom or to the antigen of the venom, then you're going to get clonal response happening, clonal selection, where it will produce copies of itself by mitosis so that they are identical. Those beta cells will then be able to produce plasma cells and memory cells. The second dose will be able to produce the antibodies in a higher concentration and quicker because you will already contain those memory cells. So the first dose must be small so the animal isn't killed. It does activate their immune system, but we don't want them to be harmed. So they will allow the idea for marking point 1, the context of primary or secondary immune response. They'll allow the idea if it's given in relation to T or B cells. They'll allow a description for specificity, so if you're talking about them being complementary to each other. For clone, they will also accept reproduced by mitosis. Clonal selection of B cells would get you marking point 1. For marking point 3, a lot of antibodies for a higher concentration of antibodies is also okay. But it's the idea that it has to be a specific B cell that is going to be complementary, or it's going to be complementary and specific to that venom. They will reproduce by mitosis through clonal selection. Those B cells will produce the plasma cells and the memory cells. So then when you're given the second dose, you can produce the antibodies in a higher concentration and more quickly because you will already contain those memory cells.